Hello, hello! This video is about nitrogen fixation and the start of amino acid biosynthesis. Learning objectives. So after watching this video, you should be able to describe the process of nitrogen fixation, understand the organisms involved and what their roles are, the roles of ferredoxin, dinitrogenase, and the nitrogen reductase, and the purpose of Leghemoglobin in relation to nitrogen fixation. You should also be able to draw the two enzyme pathways for incorporating ammonium that start amino acid biosynthesis, and then list the steps for proline biosynthesis. Since there's a whole bunch of amino acids, amino acid biosynthesis doesn't follow a nice linear step-by-step pathway of biosynthesis like the fatty acids did or glucose did through gluconeogenesis. Instead, it's kind of scattered all over the place. And so I wanted to do a really big picture just to get an idea so you can see where we're going today and then where we're going to be going in the next couple of videos. And I also want to note that we're only going to be looking at amino acids that are considered non-essential. Amino acids that are considered non-essential are the ones that your body will synthesize. And so that's why we're going to be going over those metabolic pathways because they exist. The essential amino acids are ones that you must eat. We don't have synthetic pathways to make them in the body, so you have to eat those. So obviously we're not going to go over those. We're only going to do the synthesis of the non-essential amino acids. So I'm going to go ahead and erase that. Doodle-doo. And I've got a big picture diagram. I'm going to zoom myself out and up a little bit. I'm going to start at the top and work my way down. And the carbon skeletons for the amino acids come from glycolysis and TCA cycle intermediates. So I'm going to draw an overall diagram highlighting the key carbon intermediates that give rise to the different amino acids. So again, like I said, I'm going to start at the top and go all the way down. It's not a particularly big picture or not a particularly big drawing. So again, I think you could probably fit this on one piece of paper depending on how big you draw. All right, let's start at the top. So one of our main carbon intermediates is glucose. And starting with glycolysis, a few steps into glycolysis, we get to this intermediate called glycerate 3-phosphate. I'm going to do 3P for phosphate. Glycerate 3-phosphate is the starting point for serine, and then serine branches off to give us cysteine and glycine. Okay, so those are three non-essential amino acids. Again, they're non-essential because your body has pathways to make them. Going down from glycerate, a few more steps into all the way to the end of glycolysis, we find ourselves with pyruvate. And pyruvate is only one step away from alanine. Okay. And continuing on, pyruvate can be made into acetyl-CoA in one step, the pyruvate dehydrogenase complex. And then from there, we have the TCA cycle. So you've got OAA combines with acetyl-CoA to make citrate. I'm just trying to get all the key intermediates here. A couple steps down in those are supposed to be dots. In the TCA cycle, you get the intermediate alpha ketoglutarate, and then it's several steps to get back up to OAA. All right, from OAA, we can get to aspartic acid. I've actually already seen that reaction. And from aspartic acid, you can get to asparagine, ASN, and then you can also get to threonine. I'm going to make that go away. I'll go ahead and box those. Okay. And finally, off of alpha ketoglutarate, we can get to glutamic acid, something we've also already seen before. From glutamic acid or glutamate, we can get to proline. And from glutamic acid, we can get also to GLN. I'm going to go ahead and box those. So what I've done is I boxed all of the amino acids. So again, these are all the amino acids that we're going to go through. We're going to make all of these over the course of the next videos. And the focus of this video is actually going to be to start with nitrogen from the atmosphere, reduce it to its functional form, or not its functional form, its reactive form, NH4+. And that can be incorporated to make, it can be incorporated here to make glutamic acid. It can also be incorporated here to form glutamine. It's a really interesting process to go from N2 to NH4, and so we're going to spend quite a bit of time on it, but just as an outline of what's coming up. So today's video, we're going to be here in pink. This is what we're going to cover. The reduction of N2 to NH4, which is called nitrogen fixation. And then we're going to make 3 amino acids from there, glutamic acid, proline, and glutamine. And then we're going to have another video, a second video that looks at these ones over here, including this one. And then our third and final video, here's I'll do yellow, is going to look at making serine, cysteine, and glycine. That's the really big picture, okay? Really, really big picture. Again, here today, we're going to start with N2 from the atmosphere, make it into its ammonia or ammonium, and then incorporate it really, really fast as fast as you possibly can, because this is really, the ammonia is really, 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 really toxic. So it needs to be incorporated really, really quickly into molecules so that it doesn't kill the cell, essentially. All right. A little bit about N2. N2, which is the most abundant molecule in the atmosphere, is incredibly unreactive. And that's because it's got a triple bond between the 2 nitrogens. It's just really, 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 really hard to break. But it's the most abundant thing in the atmosphere. And it's the two more reactive forms of nitrogen are either the fully reduced form, NH3, so fully reduced, has all the hydrogens, or the fully oxidized form. Either of these two forms is just more reactive. We're going to make this one. What do I want to say about this? I'm going to say biologically useful nitrogen is extremely scarce. So there's this saying in biochemistry that you can be surrounded in nitrogen but starving from nitrogen because you're constantly breathing in and out, like as a human, you're constantly breathing in and out nitrogen gas, but your human body can't do anything at all with nitrogen gas. It just exhales it right back out. And the most biologically useful form of nitrogen is NH3, the reduced form, and humans don't make it at all. In fact, bacteria are the primary makers of NH4, NH3. So let's talk about that just a little bit. So we're going to spend, what am I trying to say? There's some really cool reactions that happen in these organisms that are non-humans. So bacteria living symbiotically with plants, and we're going to talk a little bit about the enzyme that does this, because it's so cool. Humans do not have this enzyme, and we would not survive. We would not be on this planet without that enzyme. It turns out that the reaction of NH2 dinitrogen all the way to the reduced form is actually thermodynamically favorable. So if you remember what that means, if this is, I'm just put an E for energy, and this is reaction progress. If I put the reactants here into H2, if this is thermodynamically favorable, that means that the process itself has a negative delta G. So NH3 is here. Remember, delta G is a difference in energy between the reactants and the products. Here, this is negative delta G. Well, what makes N2 so hard to work with is the fact that, and I'm going to be super dramatic about this, it has incredibly high activation energy. So in order to get over the activation energy to get to NH3 is really, 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 really hard to do. There is one industrial process that overcomes this high activation energy, and it's called the Haber-Bosch process. It's a really famous process for reducing nitrogen or fixing nitrogen. It comes at a huge cost, though. This Haber-Bosch process, which is an industrial process, so it happens in a factory, happens at really, really high temperatures, 400 degrees Celsius, which I did a quick conversion for you. That's 752 degrees Fahrenheit. Super high temperature and a really, really high pressure, 300 ATMs or 3,000 PSI. For reference, the pressure in your tires on your car is probably about 32 PSI. Extremely high temperature and high pressure to get over that the activation energy barrier. It's very environmentally harsh process. It's extremely environmentally unfriendly. And biochemically, cells cannot do this. Cells cannot increase their temperature to 752 degrees Fahrenheit, and they certainly cannot increase their pressure. They would explode in either direction. They would unfold, they would explode. Cells cannot do this. So what do cells do? What does biology do? What do we do in biochemistry when there's an incredibly high activation energy bump? How does it come down? We use an enzyme in biology. And it's a very special enzyme that we call the nitrogenase. And it has two main subunits to it, but overall it's called the nitrogenase. The nitrogenase doesn't live in all species. It only lives in species that are categorized as diazotrophs. Flashcard time. And diazotrophs can be either bacteria or archaea. So the dinitrogenase or the nitrogenase, I'll just call it the nitrogenase, can only be found in some bacteria and some archaea. If it is there, then those organisms are called diazotrophs. No eukaryotes can house a nitrogenase. So plants don't have one, humans don't have one, your dog doesn't have one, your cat doesn't have one, hamsters, whatever, you name it. Eukaryote-wise, there are no eukaryotes on this planet able to do the reaction of taking N2 to NH3. But some really cool bacteria and some really cool archaea can do it, and those bacteria and archaea that can do it are called diazotrophs. Of these diazotrophs, many of them are anaerobic. That means they cannot survive in oxygen conditions. So no O2, and that's going to become important. Some of the diazotrophs are symbiotic with plants in their root nodules, and these are the ones that we're going to focus on. And that's what this diagram down here at the bottom is. So we're going to look at bacteria that exist symbiotically with plants and the little root nodules. So the rhizobia here is bacteria. And it's a bacteria that's a diazotroph. Looks like a diazotroph, which means inside of it there's a nitrogenase. And I'm pretty sure that's what the little blue on the inside is supposed to be representing, is the nitrogenase. And these bacteria are able to infect or live inside of the root nodules. All these little, I don't know, most of these bacteria that live inside of root nodules are infecting legume plants. Like, think beans. They infect the inside of the root nodules, and that is what is shown. What color should I use? Let's switch this one out to this. That's what's shown over here. So the bacteria are in yellow. The pink is the plant. Okay, so inside the bacteria is a nitrogenase, and the nitrogenase catalyzes the reaction of taking N2 from the atmosphere and making NH3. So the bacteria will give the plant NH3. And this is a mutualistic type of symbiosis, because what the bacteria get in return, so the plants get, mutualism means that both organisms benefit. So plants get the useful form of nitrogen, and the bacteria gets ATP from the plant. It gets carbon skeletons, or carbon energy metabolites. Let's call them carbon metabolites. And it also receives, how do I say this? The plant also supplies the low O2 conditions for the bacteria. So that's what each of them get. So the bacteria give the plant the useful form of nitrogen, and the plant gives the bacteria some ATP, carbon metabolites, and the low O2 conditions. And the ATP part is particularly important. I'm trying to think of like what colors can I use on top of this one, maybe blue. Because if you'll notice in here, in order to get one N2 reduced to 2 NH3s, it's going to cost us 16 ATPs every single time. So that's a lot of ATP that needs to go in to this reaction. And you know me, we're going to look at this enzyme a little bit more closely, well, because it's really cool. And I also never get to talk about plants and bacteria, so we're going to spend a little bit of time there. So let's talk, now we're going to go on and look at the nitrogenase, specifically like what are the steps for catalyzing this enzyme reaction, where do the electrons come from, who does what. All right, so let's look at the players. I'm going to move this. I got to move this over just a little bit. Let's go this one. So there's three main players here, protein-wise. There's this FDX-FLD, that's a protein called ferredoxin. Okay. And then there are the two subunits of the nitrogenase. And this one that's number 2, I kind of labeled them down here. I'm just going to call it the reductase. And then subunit number 1 is the dinitrogenase. Together, these are called the nitrogenase. All right, so just to remember, we're going to need 16 total ATPs, and we need 8 electrons. The source of electrons is pyruvate. Pick a different color. Let's go blue. All right, so pyruvate. Oh, I'm in highlighter mode. I don't want that. Bye. Blue. Yes, blue. Okay. I'm going to slide this over just a little bit. Pyruvate can give 2 electrons at a time. So that means total, if we need 8 electrons and each pyruvate is holding on to 2, we're going to need 4 of these total. Because the first thing that happens is pyruvate will pass electrons 2 at a time to ferredoxin. And this reaction that ferredoxin catalyzes is extraordinarily similar to an enzyme called the PDC. Because CoA goes in, and I'm going to do the overall. So 4 CoAs are going to go in total to combine with pyruvates, 4 of them, to move a total of 8 electrons. And that's going to leave you with 4 acetyl-CoA and 4 CO2s. Now, if you remember with the PDC would have also generated NADH as the electron carrier. That doesn't happen in this case. That's the big difference. Instead, the electrons go and they are harbored within ferredoxin because ferredoxin has these iron sulfur clusters that can hold on to electrons. And it can hold on to about 2 electrons at a time. So this process is going to repeat and repeat and repeat and repeat. It's going to repeat 8 total times. Yeah, I got that right. It's going to repeat 8 total times so that we can reduce 1 N2 all the way to NH3. All right, so pyruvate passes electrons to ferredoxin. In order for ferredoxin to be able to pass electrons to the nitrogenase, what happens is the ATP binds to the reductase. This causes a conformational change. You'll see that it has changed shape. So you get conformational change. I'm just going to do a delta for change. And then now all 3 proteins can interact. Okay, so I guess I'll do a 1. Pyruvate passes electrons to ferredoxin. 2. ATP binds to the reductase. Conformational change happens. And then 4, and there's more steps in this, but then 4, all 3 of them are bound together. Okay, so you have ferredoxin is bound, and then the reductase is bound, and the dinitrogenase is bound. And then I guess what's also shown here in number 5, or what I'll call step 5, is that electrons pass from ferredoxin, I'll just do FDX, to the reductase. Oh, excuse me. Why do I scribble? They pass to the reductase. And then those get passed to what's called the P cluster. I'm trying to think about what colors I have here. This hourglass looking structure that is also iron sulfurs is called the P cluster. And the P cluster is part of the dinitrogenase. All right. Doodle-doo, doodle-doo. All right, so electrons go. Oh, you can't really see that. I guess I'll do that one. All right, so electrons go from pyruvate to ferredoxin. ATP at the sort of at the same time and parallel happenings binds to the reductase, changes conformation. Now everybody's bound together, and the electrons are the iron sulfur clusters are all now close enough to be able to pass electrons. So electrons go from the iron sulfur clusters in ferredoxin to the iron sulfur cluster in the reductase into the P cluster. So again, all of these pieces have to be close to each other because electrons can't just jump from any number of distance. The iron sulfur clusters have to be close enough for the electrons to be able to jump. They're not, I don't know, they're not Olympic superheroes. They can only jump short distances. Okay, so what was I on? Step 5. This is an overall huge step. So now we're on the bottom half of this diagram. Whoa, 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 whoa. I was in blue. Okay. Step 6, down here. A couple things happening at the same time. ATP is hydrolyzed. At the reductase here, while at the same time electrons move to the molybdenum iron cluster, the MOFE cluster, of the dinitrogenase. And then finally, step 7, again, 2 more things are happening here. ADP is released. Which dissociates what dissociates 1 from, or 2 from 1. The 2 subunits separate. And then N2 is reduced to NH3. So N2 binds to the molybdenum iron cluster and is reduced to NH3. And then this goes, it'll go around until all 8 electrons have landed on hold on, let's change this here. Okay, so ATP, oh, I can move that? I've been able to move that this whole time? Okay, ATP binds, all 3 of these pieces come together, and electrons move through to the P cluster. ATP is hydrolyzed to ADP and PI. Electrons move to molybdenum. Now this has to, in order to get N2 reduced to NH3, you need 8 electrons here. We can only move 1 electron at a time. Oh, I meant that to be in the black color. Where'd it go? So this whole process, in order to move the 8 electrons, this whole process, this one has to happen 8 times. This has to happen 8 times, and this has to happen 8 times. So on the very final 8th round is when this whole reduction occurs. It doesn't happen every single round. It only happens once the molybdenum iron cluster has collected all 8 electrons. Now, if you're trying to count on your ATP, you're going to realize that you're short by 8 ATPs, because right now we're only showing one ATP going through every single round. And that's not entirely accurate because the reductase can bind, well, not only does it can, it does, it binds 2 ATPs every single time. So there's actually 2 of them here, 2 of them there. 2 get hydrolyzed at the same time, and 2 ADPs are released. So once this goes through 8 total times, that's where you get your 16 ATPs being hydrolyzed in order to build up really enough energy to reduce the dinitrogen into NH3. So this is another view showing the reductase and the dinitrogenase together, and also showing, you know, some of the components that I described above, where the reductase has 2 ATPs that it binds every time and hydrolyzes every single time. And the other thing I really liked about this diagram is that it really shows how close all these iron sulfur P cluster molybdenum iron clusters are together in order to be able to pass the electrons. And the note that I wanted to make here, so this piece right here is the reductase, and then that makes this chunk over here the dinitrogenase. And I wanted to come back to this idea that most of the diazotrophs are anaerobic, and that's because the nitrogenase has to be kept, the environment around the nitrogenase has to be kept in extremely low O2 conditions, because the oxygen will destroy the active site of the nitrogenase. And we're going to talk about this a little bit in class, but you might already be able to figure it out. O2, O2 being a linear diatomic molecule is very similar to N2, also being a linear diatomic molecule. O2 competes for the same active site that N2 does, and oxygen has a very high affinity for the atoms in the molybdenum iron cluster. So if oxygen gets on there, it means that nitrogen is not able to bind as easily. And basically O2 is like a suicide inhibitor of the nitrogenase. So what does it have to do with the plant? We're going back to the plant. The plant. Remember I said the plant is in a mutualistic symbiotic relationship with the bacteria. And part of the mutualism is that the plant provides some of the TCA cycle intermediates, it provides some of the ATP, but the plant also provides the low O2 environment. Okay, and the way that it does this is the plants that are legumes, so these root nodules, have this protein called Leghemoglobin. So I'm going to put the Leghemoglobin, I'll just call it Leg HB. So what you're looking at in this diagram is the bacteria is here on the inside, and it's surrounded by plant cells. So there's a plant cell here and a plant cell here. So you'll notice that a lot of the carbon intermediates, the plant is making those. Well, that's photosystem 1 and photosystem 2, but the plant is supplying the carbon intermediates. It's also keeping O2 outside of the bacteria. And the way that it's able to do this is through the Leghemoglobin. So there, I'm just going to keep the Leghemoglobin in purple here. The Leghemoglobin is a globin. Remember our myoglobins and our hemoglobins. It has a heme on the inside. And its purpose is to, I'll do Pac-Man style, is to bind up O2 in the plant cell and keep it in the plant cell so that it prevents the O2 from getting into the bacteria and competing with the dinitrogenase and destroying that dinitrogenase or the nitrogenase. And this Leghemoglobin is really actually more similar to myoglobin, but we'll compare it to regular hemoglobin. The KD for Leghemoglobin, we'll just do leg HB, is 10 times smaller than regular hemoglobin. So that means that it binds O2 really well. It doesn't give it up. Anyways, I thought this was a really fun opportunity for me to bring the globins back into your life. I know you're really excited and happy about that. Anyways, these diazotrophs that live in the root nodules are mutualistic have a mutualistic symbiosis relationship with the plant, in that the bacteria is giving the plant and we'll talk about how it's incorporating this, but it's giving the plant the nitrogen, or the molecules that are rich in nitrogen, and in return the plant gives the bacteria some carbon intermediates, some ATP, and it helps keep the oxygen environment around the diazotroph very low so that the nitrogenase stays happy. So we've made NH3. And because of the pKa of NH3, it's really NH4 inside of the cell, which is very toxic. And there are two main routes, two main pathways, two main enzymes to soak up that ammonia really fast in order for it to not be so toxic. So there's two main enzymes. One of them you have met already. We'll do in two pathways here. One of them you met here on the left-hand side is glutamate dehydrogenase. This was that step that comes right before the urea cycle, where alpha-ketoglutarate, here we're going in the opposite direction. Alpha-ketoglutarate is going in, NAD or NADPH, remember how this enzyme is unique that it can use either, will also go in to make glutamic acid. So again, this is sort of the reverse reaction from when we started the urea cycle. This one's actually I'll say this is a minor pathway. This is a minor pathway using the glutamate dehydrogenase, and that's because the equilibrium doesn't favor glutamate. It actually favors the production of NH4. So this is a minor pathway, but it is a pathway that can create glutamic acid. If you remember, we're in amino acid biosynthesis, so we've just made an amino acid, one of them. Yes, I realize it took us a while, but I thought the nitrogen story was really cool. It's a minor pathway, but it does happen. The other more major pathway uses an enzyme called glutamine GLN synthetase. Why do I never make enough space for myself? I don't know. I have this ability. Hey, man. There we go. Back to pink. GLN synthetase. I didn't actually make it that much better. GLN synthetase will take NH4 and glutamic acid and spit out GLN. If you remember glutamic acid, NH3, C, CO, two methylenes to a carboxylic acid. Well, if you throw a nitrogen on the end there, on the end of the side chain, you get GLN. COO minus. There's the backbone. The backbone hasn't changed. Two methylene groups to the carbonyl. Now we're just going to put a nitrogen on the end. That last nitrogen there came from ammonium. This does require an input of energy. ATP is needed and is hydrolyzed into ADP. and pi. All right, so we've made 2 amino acids. And before I end this, we're going to make the third one. So glutamic acid is the direct precursor for the third amino acid of the day, which is proline. And I'm going to go to the next slide to show you the synthesis of proline. Did I get everything I want to talk about there? I sure did. All right, so this is not a pathway that I am expecting that you memorize, but I do want you to see we are starting with glutamic acid at the top and we are ending with proline at the bottom. A couple things to realize that overall this costs an ATP if you're starting with glutamic acid, and it also costs 2 NADHs or NADPHs. And the overall steps are relatively, how do I want to say this? Sort of straightforward. So first, to get to here, you make an aldehyde. Here's the aldehyde right here. You make an aldehyde, and then this really cool thing happens. Look at this one, says non-enzymatic. So this step is not catalyzed by an enzyme at all. There's a spontaneous cyclization event to get to the ring for proline, and then there's another redox event to get you all the way down to regular proline. All right, so really that's what I wanted you to know, is that this is the cost of making a proline, and these are the 2 major overall steps. Make the aldehyde, and then there's spontaneous cyclization to get to the ring, and then a final reduction event to get you all the way to proline. So again, overall major recap, we started with N2. It got reduced to NH4, and that cost 16 ATPs. NH4 can be made into GLU, and you need alpha-ketoglutarate as the carbon backbone. So you throw a nitrogen on alpha-ketoglutarate. This is a redox event. And then you get to glutamic acid. Glutamic acid can be used to make proline. And this requires an ATP and 2 NADPHs to get to the proline. NH4 can also, I'll just go from GLU to GLN. GLU can combine with ammonia to give you GLN, and that's our third amino acid of the day. This also costs an ATP. That was our big overall I don't even have the word for it. That's our big picture of the day. So next time we're going to look at some other really interesting reactions, and we'll keep trekking along here and make some more of the non-essential amino acids. I will see you when I see you. Bye.